Polymerase chain reaction, or PCR, is a revolutionary molecular biology technique that allows for the amplification of a specific DNA sequence from a complex template of genetic material. Developed by Kary Mullis in the 1980s, PCR has transformed the field of genetics by providing a method to replicate DNA outside of living organisms, thus enabling detailed study and manipulation of genes. The process of PCR is relatively simple but highly precise, involving several key ingredients: the DNA template, the DNA to be copied, a pair of DNA primers, short sequences that initiate the PCR, nucleotides, the building blocks of DNA, and a special enzyme called DNA polymerase. This particular enzyme is often extracted from the bacterium Thermus aquaticus, thus the polymerase is commonly termed Taq polymerase. Number 1. Denaturation. The reaction mixture is heated to around 95 degrees Celsius, which causes the double-stranded DNA molecule to separate into two single strands, essentially unzipping the DNA helix. 2. Annealing. The temperature is lowered to between 50 to 65 degrees Celsius to allow the primers to attach or anneal to their complementary sequences on the single-stranded DNA templates. 3. Extension. The temperature is raised to about 72 degrees Celsius, enabling Taq polymerase to synthesize new strands of DNA by adding the correct nucleotides in sequence to the ends of each primer. This cycle is repeated typically 20 to 40 times, which, through the exponential increase of the DNA fragment, results in millions to billions of copies of a specific DNA sequence in a matter of hours. It allows researchers to amplify DNA segments for sequencing, identifying mutations, or studying genetic disorders. It can replicate specific genes that can then be manipulated and inserted into organisms, playing a crucial role in genetic engineering. PCR provides a tool for genetic fingerprinting, essential in forensic sciences for matching DNA samples at crime scenes to suspects. Detecting infectious diseases, it can identify the presence of pathogen DNA, aiding in the diagnosis of conditions like HIV, tuberculosis, and viral hepatitis. Cancer diagnostics. PCR can detect mutations in oncogenes or tumor suppressor genes, indicators of specific types of cancer, leading to early diagnosis and tailored treatments. Genetic disease testing. It is used to test for genetic disorders like cystic fibrosis or Huntington's disease by looking for specific genomic alterations. The COVID-19 pandemic further underlined the importance of PCR, as it was the primary tool used worldwide for the detection of the SARS-CoV-2 virus. The ability of PCR to detect minute amounts of viral genetic material made it invaluable in monitoring and containing the spread of the virus. PCR's high sensitivity, however, also means that contamination can be a significant problem, requiring strict laboratory protocols to prevent false positive results. Despite these challenges, PCR remains a definitive method for analyzing DNA and an indispensable tool in medical diagnostics and genetic research, reflecting its enduring impact on biotechnology and the life sciences.